The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You have logged on to the Nelson Labs webinar, the Reprocessing Rethought, Reprocessing Considerations During Your Design Development for Reusable Medical Devices, a reoccurring webinar presentation every other month here by Nelson Labs. Just a few housekeeping items I'd like to take care of before. If you miss one of our webinars, you can always refer back to them. And you can find them on the Nelson Labs website on the on-demand webinars page listed under the Education tab. You can also receive notifications of upcoming live and on-demand webinars by using the like button on Facebook or following on LinkedIn. We would also invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There you'll also find curated content from industry thought leaders. We would also like to mention that if you're interested in one of the Nelson Labs 3-day seminars, by attending this event, you can put in a registration code of seminars10 for a 10% discount when registering for any future seminar. So today, we are joined by Griffin Kamick. Griffin is a senior expert consultant for the healthcare reprocessing section at Nelson Labs. He graduated from Brigham Young University, Idaho, with a bachelor's degree in biology and has been with Nelson Labs since 2014. During this presentation, we do encourage your questions. Please submit them anytime in the chat box or in the questions box so we can have an open forum discussion. This Reprocessing Rethought series is intended to be more conversational with audience questions, filling the majority of the time, so please submit them at any time. And we will go up to the half-hour mark. And beginning with today's presentation, I will now turn the time over to Griffin.
Hello. Thank you for that introduction, and thank you all for being able to join this Reprocessing Rethought. I'm very excited to have this discussion with you all and to kind of talk about reprocessing validations and the important considerations to make during design development in relation to reprocessing. As Mike said, my name is Griffin Kamick. I have been with Nelson Labs since 2014 and mainly been in the reprocessing portion of Nelson Labs. And so I'm very excited to talk about reprocessing validations, talk about reusable medical devices. It's what I think about every day. And so I'm excited to have this discussion with you all. And kind of just as the previous presentations have been, these rethoughts, really this is kind of an open forum, so please feel free to ask questions about these special considerations to make during the design development portion of your medical device. To kind of start out, I just wanted to kind of give a little bit of, I guess, housekeeping, or kind of give another baseline of just what a reusable medical device is, just so we're all aware of kind of what we'll be talking about today, or what types of devices we'll be talking about today. So a reusable medical device is a device intended for repeated use on different patients with appropriate decontamination and other processing between uses. So these devices can range from invasive devices to non-invasive devices, and in some cases, non-patient contacting devices. So it's a pretty broad category. It encompasses the devices like surgical equipment and endoscope, and then it even encompasses devices like blood pressure cuffs, heart rate monitors, items that you may generally think of as capital equipment in hospitals and other healthcare facilities. And even at-home devices like CPAP masks or other menstrual cups or other kind of single patient use but reusable devices. It's a pretty broad category. So to help understand kind of how to reprocess reusable devices, the reusable medical devices are categorized into 3 classifications using what's called the Spalding classification. Again, this might be a review, but I just wanted to kind of lay a baseline for the sorts of devices we'll be talking about. And so the Spalding classification divides reusable medical devices into 3 categories, kind of based on their interaction with the patient, as well as their kind of the risk associated with these devices to the patient. So the first device, or the first classification, is our non-critical devices. These are those non-invasive devices that are either not patient contacting, or at most will contact intact skin. So again, the blood pressure cuffs, monitors, other capital equipment that may be at healthcare facilities. These are your non-critical devices, and they're typically not very complicated in design, although they still have their challenges when trying to meet the requirements for reprocessing validations, which we can talk a little bit about that during this little presentation. The next classification are semi-critical devices. These are those that will contact mucous membranes or non-intact skin. So these are devices such as endoscopes and speculums, ones that are a bit more invasive than your non-critical devices, but they're not necessarily contacting sterile parts of the body. They are often a bit more complicated in terms of design, as well as use, than your non-critical devices, but they're and they represent a higher risk than your non-critical devices. And as such, the reprocessing procedures are going to be a bit more complicated. And then lastly, the last classification we have are your critical devices. So these are devices that will contact sterile areas of the body, including blood pathway. So these are going to be your surgical instruments, devices that can be fairly complicated, have a lot of moving parts, can be difficult to clean, will probably become quite contaminated during use, whether that's with blood or tissue or other bodily fluids. They're going to represent the highest risk, or these devices kind of represent the highest risk, as again, they're coming in contact with those sterile parts of the body. So ensuring that they're clean from patient to patient is going to be very important. It's important to know where your medical device falls into this classification, because that will let you know how you're going to reprocess your device and how thorough your reprocessing procedures need to be. Your non-critical devices, those are going to be typically wiped down with a pre-saturated wipe. And then your semi-critical and critical devices are going to have a bit more involved cleaning procedures, whether that's a manual process or an automated process. And then they're going to either be high-level disinfected or sterilized, those critical and semi-critical devices. But each of these types of devices, again, comes with their own struggles and challenges when it comes to device design. And there are some common device or challenging designs to not necessarily be aware of, but be, or not necessarily avoid, but be aware of. There's items like braided cables, aluminum-based metals, pliable materials, such as silicone, rubber. O-rings represent challenging designs. Textured surfaces, hinges, springs, lumens and dead-end lumens, and just overall any inaccessible cracks and crevice. These are kind of pretty common challenging areas that may harbor unwanted organism or other organic material and may be difficult to clean. Again, it's not necessarily avoiding these designs, because these designs are here to help make your device usable. And so it's not that you necessarily want to avoid it, it's that you want to design your device if you need to use a spring, or if you need to have a hinge or have textured surfaces like knurling, to design your device in a way that will allow for effective reprocessing. And really that thought process starts during the initial design of the device. What you don't want to do is get to the very end. You've designed your device, you've done human factors testing on it to make sure that it's usable, and then you go and do your reprocessing validations, and then you find that it's quite difficult to clean and or disinfect or sterilize, and then you kind of have to go back and redesign it to make it cleanable, and then you maybe have to redo some of those other tests along the way as well. So if you keep reprocessing in mind at the beginning, during the design development, it will save you a lot of time and money and really headache at the end. It'll help you to ensure you have effective and efficient reprocessing validations, but then it also will make your device more appealing to hospitals and healthcare facilities, because it is able to be effectively cleaned by the technicians in the sterile processing departments. And so that was kind of the groundwork, or the baseline I wanted to establish and kind of go over. Really, at this point, I want to turn the time over to you all to ask really any questions about design development, or design in reference to reprocessing.
Okay, Griffin, here we go. We got our first question. Bacterial endotoxin measurements are required for medical devices, but it doesn't appear to be a requirement for medical instruments. Is bacterial endotoxin measurements on medical instruments required? If not, why not, given the risks associated with endotoxins?
Yeah, so that's an excellent question. So for reusable medical devices, bacterial endotoxin testing is no longer recommended per AAMI ST98. So that's the cleaning validation standard for here within the US. And that does not list bacterial endotoxin testing as an endpoint during your reprocessing validations. Now, I believe ISO 15883-5, which is where you're going to get a lot of information for cleaning validations when you're submitting outside the US. It still lists bacterial endotoxin testing as a possible endpoint during your reprocessing validation or your cleaning validation. But if you're looking to market within the US and outside the US, I would most likely not do bacterial endotoxin testing, as it's not a requirement. It's not seen as an applicable endpoint during cleaning validation testing. Now, for single-use devices, it still is an important endpoint that needs to be tested and evaluated. Whereas for reusable devices, in terms of the cleaning validation, it's not viewed as an applicable endpoint, and other endpoints like protein or hemoglobin or total organic carbon are viewed to be a bit more applicable in reference to residual contamination from patient after the cleaning process. Now, it is important to note that there are other documents out there that are meant to kind of monitor things like the water quality used during reprocessing in hospitals. So documents like AAMI ST108, so that's the water quality standard for hospitals and healthcare facilities. Within that standard, you'll find that there is a requirement for bacterial endotoxin levels within the different water classifications. So utility water and critical water are the two water classifications within that document. And you'll find that there are requirements for monitoring the bacterial endotoxin level within those two types of water. And those are going to be your types of water that will then be used to rinse and prepare your detergents and rinse the devices during your cleaning validation or during or sorry, during the cleaning process within the hospital. So it is not an applicable endpoint for reusable devices, but it should still be monitored by the hospital. The bacterial endotoxin level should still be monitored by the hospital in order to meet those requirements in ST-108.
Great, thank you, Griffin, for that answer. Here's our next question. I'm designing a handheld surgical instrument. Can I expect ball bearings within the handle to pass a cleaning validation?
So it will present some challenge. I can't necessarily guarantee that it'll pass or that it'll fail. I don't want to dash your hopes. It will present a great challenge. So there is going to be a lot of areas where organisms or organic material, as was showing on the screen, is going to be able to hide and will be difficult to remove. So an easy way to kind of get around some of these difficult areas or these challenging designs is to allow your device to be disassembled. So if your handheld instrument is able to be disassembled so that those ball bearings can be more easily reached and are more accessible to the rinsing and to the detergent and even to brushing if possible, then that will greatly increase your chances of passing the validation, as well as having a safe device that's being properly reprocessed in the sterile processing departments. With that sort of device, though, I would expect to include ultrasonication during the cleaning process. So you'll recommend in your instructions for use to prepare a detergent in an ultrasonic unit and then fully immerse the device in there and allow the device to remain immersed during the sonication process for 10 to 15 minutes. Those are pretty typical time ranges for ultrasonic procedures. But I would definitely plan on having an ultrasonic step in your instructions for use. But again, if you can disassemble your device, that is going to greatly increase your chances of passing. Now, again, kind of the tricky part with that is you want to make sure that the disassembly is easy and that you have clear instructions in your IFU that are clear to follow for those technicians in the sterile processing department, that they can adequately and efficiently disassemble and reassemble that device without needing to have any special tools or equipment or having it take a long time, and so that they can clean it efficiently and then put it back together as needed.
Great, thank you, Griffin. Here is our next question. Is it problematic to get a recovery rate below 70%?
It depends. Now that's that's the answer for everything, I think. It depends, right? 70%. So AAMI ST98 and ISO 15883-5 do recommend that the recovery efficiency be above 70%, but it's not a strict requirement. It says something to the effect that recovery efficiencies are generally above 70%. So if it's below 70%, I think that's okay, and it kind of depends on a few things. I think it would, if you are below 70%, just an evaluation of the extraction method used. It could be that the extraction method used, while not the most efficient, was the only one available based on the design of the device or the size of the device. The device could be so large that perhaps something like swabbing is the only available method. In that case, swabbing is not often the most efficient extraction method, and so it's not uncommon to see recovery efficiencies below 70% when using that method. The other thing I would take a look at is just consistency. So if you've done more than one recovery efficiency, does it seem at least consistent between the recovery efficiencies? And if it does, then I think you're I wouldn't necessarily worry too much about it. The important part is with cleaning validations, we do apply a correction factor based off that recovery efficiency. So even if it's below 70%, we're still taking into account that recovery efficiency into the test articles and the negative device controls. It's still taken into account with those results. So while it's not, I don't think it necessitates a retest right away. If you're below 70%, I would just investigate it a little bit. So again, look at the extraction method. That's going to include, again, what was the container used, what was the fluid used to extract the device. If your device disassembles, was it extracted disassembled or assembled? Which if your device does disassemble, it should be extracted disassembled. And then, again, look at the consistency if you have enough data points to evaluate that. So if you have more than one positive control and more than one recovery efficiency, how consistent is it?
Great, here's our next one. What is the requirement you generally advise targeting for the dryness of a device after cleaning and before it goes into steam autoclave?
So in terms of validation testing, there's currently not a requirement out there. So there's no dry time validation for after the cleaning process to ensure that it's dry before the sterilization process. However, the device does need to be dry. In practice, you do need to have instructions in your IFU that will dictate the end user to dry the device efficiently and sufficiently so that it's not wet when it goes into the autoclave or the steam sterilizer or whatever the terminal process is, because that's going to impact that terminal process and it's going to impact your device. So really, in your instructions for use, you just need to have language that dictates that the device is dry before packaging it or before the next step, whatever that next processing step is, whether it's disinfection or sterilization. And then also allow the end user the needed methods to dry a device. So allow them to use, those lint-free cloths or cloths to wipe it down, or to use filtered pressurized air to dry out those lumens or those cracks or crevices. And that's typically what we see during reprocessing validations, is either of those methods. But again, there's no validation currently, or there's no requirement that you need to show or that you need to do a dry time validation after the cleaning validation or after the cleaning process. But it's still important to have language in your IFU to guide the end users.
Okay, on to the next one. Which methods could be used to evaluate the cleanability or disinfectability of a device during the development of the device?
Okay, so yeah, which methods could be used to determine the cleanability of your device design during the design development? That is an excellent question, because that's the hardest part, is because you are in the design phase. You may not have the physical device, so it may not be possible to do some sort of feasibility test to determine if it's cleanable, to determine its cleanability, or if it can be disinfected properly. And so that's where, just kind of referencing this list, you can also look at these reference documents. So AAMI TIR 12, Section 4, ISO 17664-1 for critical and semi-critical devices -2 for non-critical devices, Annex C of that document. And then Appendix E in the FDA guidance on reprocessing medical devices. These documents give lists of difficult to clean areas. And so while you're designing your device, I would definitely reference these, the portions of these documents, have them easily accessible or readily available, so that as you're designing the device, you can cross-check it against some of these design features that are listed in these documents, and you can get an idea of how difficult your device is going to be able, or how difficult it will be to clean the device or disinfect the device or even sterilize the device. And really, I would just do, you could create a table where you list the different design features, and then you can just mark an X by each one that your device has. And essentially the more X's by design features, the more difficult it's going to be to clean. And really what that means is, at that point, you need to start thinking about how involved or how in depth does your reprocessing procedures need to be. As you have more of these design features that are difficult to clean, you're going to have a longer cleaning process and a more involved cleaning process. You're going to be using more cleaning tools like brushes and syringes or flushing instruments to flush the lumens. You'll be using ultrasonic units, and you'll be using different types of brushes. So not just toothbrush style toothbrushes, but also lumen brushes and different. If you have different sized lumens, then you'll have different lumen brush sizes that you'll have in your IFU. And so really, at that point, you can also just take a look at, are some of the features necessary? And if they are, then, obviously leave those on the device. If they're not, then maybe figure out a way to, limit some of those challenging features that are on the device. Or kind of going back to the previous question, assessing whether you should make the device be able to be disassembled. Now, you don't want to make if you have an instrument tray, you don't want to disassemble every device, and not every device needs to be disassembled, but your more complex tools or instruments in that tray, maybe think about making them disassembled, or making it have open spaces so that things can flush through them, rather than enclosing spaces, making them open so that water and detergent can readily get into those hard to reach areas, rather than having it enclosed. But that is an excellent question, and that is the hardest part, is how can you determine cleanability? There's no clear answer. I would just look at these reference documents, and as you design the device, just keep an eye on, as you add a feature or as you are designing it, just keep these different lists in mind. And, as you accumulate more on your device, just know that that's going to impact your instructions down the line.
Okay, Griffin, we're going to try to squeeze in one more question here.
Perfect.
It says, what is the most popular, I'm assuming, markers for reprocessing validation? And is there any changes in Annex 1 updates that we have to change in the process required?
So for the first part, if I understand correctly, kind of what are the endpoints for a reprocessing validation?
The most popular markers.
Yeah, the most popular markers. So really, protein is going to be the most popular marker. So if you're doing a reprocessing validation and you need to do endpoint testing to look for that residual test soil, protein will most likely be your first marker or first endpoint that you'll test for, and that's going to be that way across the board. Protein is generally the most accepted one and the one that everyone wants to see. After that, it's really going to depend on your device and where it's used. Typically with surgical instruments where there's going to be more blood contact, we typically see hemoglobin being the second marker. And then for the devices that are coming in contact with mucous membranes or maybe non-intact skin or intact skin, we see total organic carbon being the second marker. But really, for surgical instruments, you can use total organic carbon as the second marker. You don't necessarily have to do hemoglobin. You can do total organic carbon. Really, you just need to select at least two, and the first one needs to be protein, and then the second one, I would generally choose hemoglobin or total organic carbon. You can do carbohydrates if for whatever reason hemoglobin and total organic carbon are unavailable. Carbohydrates can be chosen, although it's less popular. So.
Okay. And we have many more questions, Brandon or Griffin, but we'll get those over to you, and Griffin will be able to reach out to you individually with those questions. We would like to thank Griffin, our presenter, for his insights, and thank you, the viewer, for attending this session. We hope that you found it to be a valuable experience. We hope that you tune in December for our next session of the Reprocessing Rethought. Thank you, and have a great rest of your day.
Thank you.